Today we continue answering textbook questions from the Oxford IB course companion that you see on your screen with the first assessment in 2025. Today we are going to answer questions 1, 2 and 3 which are found on page 23 of your course companion book. Starting with the first question, in Homo sapiens, the smallest chromosome and therefore the shortest DNA molecule is the Y chromosome with this huge number of base pairs. The human genome has some billion base pairs in total. What percentage of this does the Y chromosome contain? You've got the two numbers here, whole genome base pairs and the base pairs found on the Y chromosome. No additional information is needed, as like for example that the shortest DNA molecule is the Y chromosome. Yep, it is. Remember, we've got in humans we've got 23 pairs of chromosomes. The sex chromosome pair consists of the X and the Y. We usually classify them as the 23rd pair. Right, in this one here we've got the number 57,227,415. So you divide this number by the number 3.08 billion. Okay, millions and billions. Use your calculator and the answer that you will get here once you do this would be 0.0185. Now multiply this by 100% and you get 1.85%. So this is your answer for the first question. The second question is the following. The bacterium Carsonella ready. Let's bring this to the whiteboard. In this question, you've got lots of information provided for this bacterium species, like the number of base pairs in its genome, the number of genes in each genome, and how many of these genes code for proteins. We are asked to calculate the percentage of bases that are adenine, cytosine, and thymine, and we are also be given the percentage of the bases that are guanine. You can ignore all this part of the question with unnecessary information. The key thing to focus is that bacteria have, is it double-stranded or single-stranded DNA? It is double-stranded DNA. Remember, all living organisms on Earth are either organisms or microorganisms have a DNA which is double-stranded, with an exception of some viruses, some DNA viruses, which might also have single-stranded DNA. So all bacteria have double-stranded DNA. Based on the complementary base pair rule, now adenines are paired with thymines and cytosines paired with guanines. You see three lines here that represent the number of hydrogen bonds. Again, we don't need that here. So if we know the percentage of guanine bases in the whole DNA molecule, actually the DNA molecule of the bacterial species is a circular DNA molecule, circular double-stranded DNA molecule, and you know that guanine percentage is 7.3%. That also means that this is equal to the percentage of cytosine, which is 7.3%, add these two together, they make 14.6%. And if you subtract this out of the total 100%, which is the whole DNA genome here, the whole DNA molecule, it's 85.4 is the rest of it. The rest of it is adenine and thymine together, 85.4%. So what you just need to do then is divide 85.4 by 2 and you'll get 42.7%. Why do we divide by 2? Because A pairs with T, so the percentage of each will be equal and half of 85.4. So each one of these would be 42.7, 42.7, and you've got your answer. Third question is about the Canine-Sicko virus. We are given the genome of this virus. Pay attention to this one. It's not given in base pairs, it's given in just bases. It contains just 2 protein coding genes, of course. The type of virus has a protein code that is only 17 nanometers in diameter. Yep, it's a tiny a virus. Its genetic material is single-stranded DNA. So this is the exception that we've been talking so far. Some of the DNA viruses are single-stranded, but not all of them. So just one advantage and one disadvantage of this DNA being single-stranded. This is a 2 marks question. So you just need to provide one advantage and one disadvantage since this is a 2 marks question. Best for you to add another one just in case the first one is not correct. Maybe make like 2 points for each of these for the advantages and disadvantages. First of all, you've been given also the diameter of the protein code of the virus. That means the virus is tiny as we would expect. And think of the single-stranded DNA as narrower. And because of that, it takes up less space inside the protein code of the virus. Double-stranded DNA is 2 nanometers wide, so a single-stranded DNA would only be 1 nanometer wide. Okay, a disadvantage here is that the bases are more vulnerable to chemical attack when unpaired, so we've got more mutations. So single-stranded, no base pairing. The DNA sequence looks like this. And this is just an example here. Okay. It's vulnerable to chemical attacks, and when the molecule is unpaired, we've got more mutations. A disadvantage for the organisms that this virus infects, as because of the mutations, it can change the proteins that are expressed. It can escape the immune system response, presenting itself as a new virus every time that infects us. You don't need to get into too much detail here, so just say, since there's no base pairing, it's more vulnerable to chemical attack when unpaired, so more mutations can be found in the single-stranded DNA.